And we are live. Wait, are we there yet? Yeah. So hello, everyone. Welcome to today's Neurotheory Forum talk in its online format. This seminar series is organized by Tim Vogels from IST Austria and Andrew Sachs from the University of Oxford, UK, and occasionally hosted by their colleagues like today. I'm Everton Agnes, project leader at the Biocentrum, University of Basel in Switzerland. And today I have the pleasure to host Professor Jeehyun Kwag. Jeehyun is a professor at the Department of Brain and Cognitive Engineering, Korea University. Professor Kwag started her scientific career in maths. She did her undergrad in mathematics at the University of Oxford, UK, but then decided to switch fields and start working with neuroscience. Jeehyun received her master's degree in neuroscience in 2004 and her PhD degree in physiology under the supervision of Ole Paulson in 2008, both from the University of Oxford. Jeehyun then did a brief postdoc still in Oxford, and at the time she was also a lecturer in neuroscience at Keble College, part of the University of Oxford. In 2010, she started her own lab at the Korea University when she was appointed as assistant professor. Jeehyun's research combines experimental work and theoretical modeling to help elucidate the brain's mechanisms responsible for information processing and memory formation. And more recently, Jeehyun started exploring the role of different inhibitory neuron types for learning and coding, with really interesting insights from the combination of experiments and modeling, which we will hear a bit more today. So before we start, just a few reminders. Use the chat as much as you want. It's at the right. And if you have questions that you like to be answered during the talk, write them there and I will try to ask them. And additionally, there is an ask a question box at the bottom of the page. Use it to post questions that you like addressed at the end of the talk. And also we'll have a Zoom meeting with Jeehyun after the talk for further discussions. I will paste the link for this meeting on the chat. With no further ado, Jeehyun, the online stage is yours. We'll focus your slide. Yeah.
Okay, thank you very much for a kind introduction. And also, this is my great honor to be invited to speak at the Neurotheory Talk Forum. So today, and also thank you very much for the great introduction. I hope I can fulfill the expectations of what I'll be presenting today. So the title of my talk today is Inhibitory Neural Circuit Mechanisms Underlying Neurocoding of Sensory Information in the Neocortex. So the title is very grand, but you will see, oops, sorry, that the screen is not fully showing, is it? Should I start again?
Yeah, it was. Now it's back.
Okay, thank you. So this talk will be covering part of the neocortex, barocortex, and how the barocortex processes information using different patterns of spikes and how those spike patterns are arising from different interneuronal networks. So as Everton kindly introduced, I am currently in Korea and I studied in University of Oxford beforehand coming here. And there I was a pure in vitro brain slice electrophysiologist. And after coming to Korea, my lab has turned into studying the computational network mechanisms of the information processing, especially the neural circuit mechanisms underlying neurocoding, how the spike trains are encoding and decoding information for sensory information processing and memory processing. So this is our very hardworking students in the lab, and we are currently investigating how different types of interneurons are working together to support the functions of the brain, and especially encoding information. So overall, we have two broad interests in the lab. One is to study the sensory information processing, especially in the barocortex. So here we monitor the activities of neurons across all layers using in vivo recordings to see how these spike patterns are coding different types of information, texture information, for example, or Whisker stimulation-evoked informations. And the other kind of a big project in the lab also is how the spikes are coding memory. So here we're also learning, doing in vivo recordings and in vitro recordings to see how spike patterns change before and after learning. And we do this in healthy brains and also in Alzheimer's brains because we think pathological brains also give us a lot of insights on how the brains might be processing in normal states. So as I just mentioned, we are using a lot of methodologies. So first of all, I started off as in vitro electrophysiologist, doing whole cell patch clamp recordings and identified neurons in the hippocampus and in the cortex. So we use optogenetic methods as well to manipulate each type of neurons to see their contributions to generating spikes and synaptic currents. Also, recently we have been getting a lot of in vivo technology methodologies in the lab, including in vivo silicon probe, recordings for recording single unit activities, network oscillations, and also calcium imaging using miniaturized microscopes to monitor ensemble activities of neurons during behavior. And now, in addition to that, we have in silico computational modeling methods adopted to complement these in vivo and in vitro experimental methodologies. So before as I said, I was mostly in vitro electrophysiologist, but after coming to Korea University, in fact, to be honest, it took almost 2 years to set up the in vitro setup, the patch clamp rig. So in the meantime, we started to do computational modeling, Hodgkin-Huxley modeling, or integral fire neuron modelings, and also trying to build out full morphology network cell models, and then extending it to small spiking neural network models to study the behavior of networks. So all three different methodologies are combined together in the lab. So in vitro, in vivo electrophysiology, plus optogenetic manipulation methods to study how the circuits, especially in our case, inhibitory interneurons, are used in processing information, and that will give us insights to the cellular and neural network mechanisms, as well as brain and structure and functions. And these data that we collect from these experimental methodologies are fed into our computational network model. So that was just show a quick short video. So we sometimes build full morphology computational models of neurons that have typical Hodgkin-Huxley type neuron spiking mechanisms, or we also have integral fine neuron models built so that we can construct realistic neuron model that can to understand the brain better, that cannot be investigated using conventional experimental methods. So now, after building these computational models, we can also confirm the experimental results, but also we can get new experimental predictions that can be tested in the experiments further. So these in vitro and in vivo electrophysiology and in silico computational modeling methods are complementing each other to give a synergistic effect in helping us find the mechanisms of how the brain works. So within the small lab, all of our students are working really hard together collaboratively to using all these three different methodologies broadly to find the answers to the questions we have in the lab. So today, I won't be able to have enough time to talk a lot about memory, but today I'll focus mostly on sensory information processing in the neocortex. And then at the end, I'll show you some ongoing projects in the lab that we're currently undertaking. So sensory information processing in the brain, we use a lot of sensory modalities to constantly explore what is around us. We hear, smell, taste, see, and touch the objects around us to make sense of the world around us. And then using all these information, we somehow store these information into memory. So in humans, we use mechanoreceptors on their skins to explore the world around the objects around us. But in rodents, they also have mechanoreceptors, but also they use whiskers to detect objects around them. And so the information collected from each whisker is sent to the thalamus, and then eventually reaches the primary somatosensory cortex of the rodent, which is also called, which comprises one of the biggest part is called the barrel cortex, where each individual whisker sends information to one specific barrel, like cortical column of the barrel cortex. So all these all together is called the barrel cortex. You can see distinct layers of 1 to 6, and then each you can see all these identifiable barrel columns where each whisker sends information to. So if you want to know what kind of information this rodent is detecting from the whisker, and how that information is encoded in the brain for the perception of that specific object that the rodent is exploring, one way to find it out is to implant an electrode. So in order to see how it is encoded in the neuronal activity, implant an electrode that penetrates all across the cortical column of the barrel cortex and records spiking activities. So this is kind of a raster plot, spike raster from other papers cited here. So these show that the timing of the whisker stimulation, and then these individual dots represent spikes when they're the timing of the spikes recorded across layer 2, 3, 4, 5, and 6. So whatever this information, whatever is encoded in this spike pattern, that must be what this animal has been detecting. So the spike patterns must be encoding the sensory information that is embedded there. So how are information encoded in spike trains? So there's a lot of different theories on how spike trains encode information. The most widely used concept is called the rate code. That is simply counting the number of spikes per given time to see, so in this case, you have 5 spikes per, let's say this box is 1 second, so you have 5 hertz of spikes. And then another neuron has 2 hertz of spikes in encoding some aspect of an object around him. And then another coding scheme is a temporal code. So each individual spikes timing might be encoding information. In this case, let's say this vertical line is the onset of the whisker stimulation, and then the first spike after the onset of the whisker stimulation might be encoding specific aspect of the object that the whisker is detecting, for example. Another code is called the synchrony code. So this is one type of a temporal code as well, but you can see that all these neurons, although they're all independent neurons across different layers, they are generating spikes almost in a similar time window. So these neurons are called synchronized. So synchronous spike timings across populations of neurons called the synchrony code. Another type of code that's mostly studied largely in the hippocampus is called the phase code, where each spike timing relative to the ongoing oscillation can also code information. So now if you go back to the spike raster plot, what do you see? You see a lot of neurons across the cortical layers are synchronized together, making up a synchrony code. And then within the synchrony code, synchronized spikes, these neurons have specific precise spike timing. So there is a temporal code. And also if you take a mean of these number of spikes per given time, you have a different spike firing rate code for individual neurons. So you can see that within the synchronized spike timing window, there is also a temporal code and the rate code. So overall, we have this idea that these neural codes exist, but in fact, these neural codes are not mutually exclusive, but they are coexisting. But generally, people study these neural codes independently. So our question is, so there is this synchrony code. The neurons are synchronized together, and within the synchronized activity, the individual spike timing exists, and overall there's a rate code. So what is the neural circuit mechanism supporting the multiplexing of neural code using many different kinds of coding scheme altogether within a given spike train for sensory information processing? So what is a how are these spikes spatial temporally synchronized across time? Across like the time and space, is there a neural circuit mechanism for this? Because if this is important for information encoding, there must be some network involved. So if we look at the barrel cortex, 80% of the neurons are excitatory neurons. So generally speaking, this is a canonical model, but if you stimulate a whisker, the whisker stimulation-evoked spikes will go through the thalamus and eventually reach the layer 4 of the barrel cortex. And then canonically, classical model tells you that it flows to layer 2, 3, and then layer 5, and then it goes to layer 6 to the serving as a cortical output. So as I said, this is a canonical model. There are also experimental evidence that from the thalamus, input directly goes to layer 5 or 6, but canonically, this is a normal flow of sensory information across the cortical column. So now, in addition to excitatory neurons, there are remaining 20% or so of the neuronal population in the cortex are inhibitory interneurons. And then these inhibitory interneurons, there are many different types. You can see, this is one example cartoon showing the excitatory neuron and then the different types of inhibitory interneurons around them. And you can see there are some PV or somatostatin or VIP neurons, or there's a CCK interneurons. They all have different morphology, different sites of synaptic context, and then different locations within the cortical column. And because there are so many, it's very difficult and so diverse. It's difficult to study the functions of each individual neuronal types, but broadly, for at least for my talk, we can divide these neurons into PV and SST interneurons, depending on the type of the proteins they express. And also you can see that these neurons have different synaptic targets of the excitatory neurons. So here you see PV interneurons are distributed all across the cortical 6 layers, and also somatostatin interneurons also are distributed across the cortical column. So these interneurons must have some sort of a function in sensory information processing, especially in processing these different types of neural codes. So generally, as I said, a lot of people only concentrated on excitatory neuronal responses from the Whisker stimulation, but recent studies show that these PV and SST interneurons also are responsive to Whisker stimulation. So these PV interneurons, in this case, in the layer 5 or in the layer 4, SST interneurons in layer 4, when they recorded these activities during Whisker stimulation, across different layers, excitatory neurons show very distinctive Whisker stimulation evoked increase in firing rates, but also PV and SST interneurons across the layers also show increase in firing rate. So this means that upon the sensory stimulation from the Whisker, not only excitatory neurons, but PV and SST interneurons are also activated. So what are their function in generating these phase, rate, and temporal codes within the synchronized activity in the brow cortex evoked by Whisker stimulation? So our question is, how do PV and SST interneurons support the generation of these Whisker stimulation evoked synchronized spike activity that contains rate and temporal code? So in order to address this question, we use in vivo electrophysiological methods of recording the spike activities across the whole 6 layers. And also we use in vivo optogenetic methods to perturb these PV and SST interneurons to see how these neural coding scheme changes. And also we use the computational in silico neural network model in order to see the dynamic changes in the network that cannot be seen using the conventional computational model. Using the computational experimental methods. So first, as I said, we are going to now record the Whisker stimulation evoked spikes in vivo. So in order to do this, we place a 32-channel multi-electrode probe into the barrel cortex, and then we stimulate the Whisker. Now, this whisker stimulation, instead of at the time of these experiments, we wanted to just, the whole purpose was to generate reliable spike patterns across all 6 layers. So we bundled up all the whiskers together and then stimulated them at 12 hertz for 3 seconds. So this allowed us to generate spikes ranging from 0 to 40 hertz, very random spike patterns across all layers. And then we recorded them using this silicon probe, all the spike patterns of all the neurons across the layers. So you can see there's a lot of neurons being recorded. But now from here, we filtered out only the excitatory neurons because we wanted to see how excitatory neurons are generating these synchronized spike timings at different frequencies. So we filtered out inhibitory neurons and then only included excitatory neurons for further analysis. And also we selected the neurons that are only showing whisker stimulation responsive increase in firing rate. So these PSTH shows that upon whisker stimulation, these neurons are showing increased firing rate. So only these neurons were included for further analysis. So now if you filter out the neurons that are only responding to the stimulation, from all these many neurons, you only have left with 12 neurons. So whisker stimulation responsive excitatory neurons are now here, plotted here in the raster plot, and then a lot of the layer 2-3 neurons are taken out, and also only one, in this case, layer 4 neurons survived. And then there are many layer 5 and layer 6 neurons that were responsive to whisker stimulation. So using this spike raster plot.
Can I ask you something before you continue? You said that you had 11 neurons out of how many? How many are you recording at the same time?
Probably this is in the order of almost 50, 60, or 100. It depends on the experiment. But generally, the responsive neurons over a population is around 20, depending on the success rate of the recording as well. But I'll show you data from other at the end of the talk as well. There are generally 10 to 20% of the neurons are responsive to whisker stimulation.
Okay. Oh, that's a big proportion.
So that reduces a large number of neurons that are further analyzed in our study.
Okay. Thanks.
So here, using these responsive neurons to whisker stimulation, now we wanted to see how these neurons are synchronized together. So this is one batch of synchronized neurons, and they're having different spike timing and spike firing rate. So we wanted to quantify the synchrony between the spike pairs between layer 4 and layer 5 and layer 4 and layer 6, because as I mentioned earlier, we're considering the canonical model of the information flow in the sensory cortex, and we wanted to, in the barocortex, we wanted to compare the input from the thalamus and then how these inputs are synchronized to the layer 5 or layer 6. So we are doing pairwise coherence analysis, right, to layer 4 and number 1 and layer 4 spike and number 2 and L5, etc. So we are doing that to measure how synchronous these two spike trains are, and these scores are shown here. So this is a spike time coherence score for each neuronal pair with layer 4 and layer 5. So you can see that these scores are taken from the maximum of the spike time coherence score, and then we plot it, and you see that some neurons, like this one, this spike train is not coherent at all. It doesn't fall within this is a plus minus 10 milliseconds of the spike train, the spike timing of the layer 4 neuron, and you can see that this neuron does not fall within any of these gray windows, but then the other ones generally fall within. So not all 3, but 2 or 3 or 1. So you see that these ones have high spike time coherence scores, and those that follow out have low coherence scores. So now for further analysis, now we take out the ones that are not synchronous, and now we are left with 9 or 10. So here we have further analysis on how we want to now see how these synchronized spikes are synchronized also for spike firing rate or spike timing as well. So in order to analyze within the synchronized spikes, whether the spike trains in layer 4 and the other layer 5 and 6, are synchronized in spike firing rate, we calculated the instantaneous firing rate. So basically, this is the reciprocal of the interspike interval of these neuronal spike timings. So if this, for example, this one has a interspike interval of maybe 4 to 12 hertz, and this one has an interspike instantaneous firing rate of 0.5 to 4 hertz. And now, if you compare these colorings, you see that these spikes, all the spikes, have similar instantaneous firing rate with the layer 4 input, and these ones have similar firing rate as the input, whereas these ones don't have the same firing rates. So just by looking at this color plot, you can see whether the spike trains in other neurons in other layers have similar instantaneous firing rate or not. Now, in order to see whether the spike timing of the neurons in other other layer 5 and 6 neurons in layer 5 and 6 are also synchronized in spike timing, we introduced a measure called the instantaneous similarity, which calculates the synchronization of each spike times of 2 spike trains. So basically, similar concept. Now, if you see the red here, that means the similarity of these 2 spikes having a similar spike time is very high. So red indicates high similarity, and yellow indicates low similarity. So if you see a lot of red, that means this neuron for this specific spike train or this spike ISI, well, they have high similarity. So this sounds all complicated, but so far we have only isolated the Whisker responsive neurons that are excitatory and that are also have high spike time coherence, so they are synchronized. And within the synchronized neurons, we're analyzing how spike times and firing rates are changing. So in order to do that, quantify much better, we come up with a new plot, which we call iSR-IFR profile. So here, this x-axis is the instantaneous firing rate of layer 4 input, and y-axis is the instantaneous similarity called ISR. So this is ISR as a function of IFR, and now you can see for different frequency ranges, you can see how the spike times are similar for the synchronized neuronal pairs in layer 4 and layer 5, and layer 4 and layer 6. So here, this graph is a horizontal line, which shows us that in this control condition, when we are moving the whiskers in our experiments, the spike times are synchronized across all frequency ranges, ranging from 0.5 to 50 hertz. So this profile is the one you need to remember to follow the other slides coming. So this basically means that layer 4 spikes are synchronized to layer 5 neuronal spikes in spike timing and firing rate in the within the synchronized packet of spikes. So now what we wanted to understand, so this is a summary again, Whisker stimulation-evoked spike trains in layer 4 and layer 5 or layer 4 and layer 6 are synchronized for synchrony code for all spike times, which is called the temporal code, and across all frequency ranges, which we call the rate code. So this is what we have. So now we wanted to see the contributions of PV and SST interneurons in generating these synchronized spike times over different frequency ranges. So in order to do that, we use optogenetic manipulation methods to perturb the network, to bias the network to have more PV activation or less, and similar for SST. So in order to do that, we use optogenetic methods. In the PV Cree or SST Cree mice, we injected channel rhodopsin or ARC rhodopsin to either activate PV or SST interneurons or inactivate or silence PV and SST interneurons. So using blue light in neurons that are expressing channel rhodopsin, if you use a blue light, the PV neuron will generate spikes, and likewise for SST interneurons. And also if you express arc-rhodopsin to PV or SST interneurons, and if you shine a green light, these PV or SST interneurons will be silenced. So using this methodology, we will see how PV and SST activation and inactivation will change the iSR-IFR profile. So we injected this AAV virus carrying tenor-rhodopsin into the barrel cortex, and then we see that these in the PV-Cre and SST-Cre mice, and these expressing tenor-rhodopsin across all layers of PV neurons and all across the layers, as well as SST. And now when we shine a light, we can see that some PV neurons are responsive to blue light, showing increased firing rate, and SST neurons are showing increased firing rate as well in SST-Cre mice. So this confirms the successful expression of tenor-rhodopsin in PV-Cre or SST-Cre mice. Now, using these mice and optogenetic activation of PV interneurons, we repeat the same experiment again as I just showed you in the control experiment. So now we activate the PV interneurons during Whisker stimulation. We give the light slightly beforehand as well, before the Whisker stimulation onset, just to make sure that the PV neurons are already activated during the Whisker stimulation. And now this did not change the shape of PSTH itself, but now when we look at the spike train patterns in more detail, and then also analyze for iSR, IFR, and iSR, we see that the patterns are slightly different. And then when we now plot this into the iSR-IFR profile, before that, we saw a complete horizontal line in the iSR-IFR profile in the control case, but now activation of PV interneuron completely changed the profile so that the high frequency spikes are gated. So the synchrony of spike timing at high frequencies, in this case, frequencies above 12 hertz, are suppressed, whereas the spike time synchronization of spike times at frequencies below 12 hertz remain the same. So now if you do the same experiments for activating SST interneurons, with same analysis, now you can see IFR, ISR are different already. And then these high firing rate spikes have red colored here. So which means that activation of SST interneurons gated the synchronization of spike times at low frequency, low firing rate for below 12 hertz, and above 12 hertz, the synchronization was intact. So you can already see that these two graphs, this graph and these graphs, are very complementary. And these effects of PV and SST interneurons gating the synchronization of spike times at different frequency ranges were not dependent on the amplitude of the light stimulation that was being delivered. So this means that this is a very robust, robust and can be generalized over different light stimulation intensities. So now we saw that PV activation and SST activation gated the synchronization of spike times at high and low frequencies. So if that is possible, now we wanted to see whether inactivation of PV or SST interneurons can promote the synchronization of spike times at the other frequencies. So in order to do this, we now expressed arc-rhodopsin to other PV or SST interneurons in PV and SST-Cre mice across, and you can see that these are expressed well across the 6 layers. And also we were able to identify some single units which had a spike firing rate suppressed upon the shining of the green light. So this shows that these arc-rhodopsins were expressed successfully across the neurons in the barrel cortex. Now we repeat the same experiments again, now silencing the PV interneurons. And now what you see is the profile is again changed. And now inactivation of PV promoted the synchronization of spike times at high frequency, firing rate above 12 hertz. And now similarly, when we inactivated SST interneurons, now that changed the iSR-IFR profile so that the below 12 hertz, low frequency spikes times were synchronized. So these were promoted. So, and this also could be generalized for all the light stimulation intensities that we use for the experiments. So these also showing that it is kind of a robust effect. So even a activation of small subsets of PV or SST or inactivation of small subsets of PV and SST has the same effect in the changing the iSR-IFR profile. So far we have this experimental data showing that activation of PV gates high and activation SST gates low firing rate spike timing synchronization, whereas inactivation of PV promotes high firing rate spike time synchronization, and SST inactivation promotes low firing rate spike time synchronization. So these two have very complementary roles at gating these synchronization and spike time, and likewise for inactivation of PV and SST. But now, if you look at these two together, they're the same. And if you look at these two iSR-IFR profiles, they are the same as well. That's quite striking. So from here, you can see that when we activate PV interneurons, that might somehow in the network cause SST interneurons to be inactivated, because so these are occurring simultaneously. That's why when we inactivate SST, it has the same effect as activating PV. And also inactivation of PV accompanies activation of SST. So these two activation and inactivation of PV is happening in vivo simultaneously. So we can infer this from these iSR-IFR profiles. So this means that these PV and SST interneurons are dynamically recruited during the sensory information processing in the barrel cortex to gate or promote the synchronization of spike times at different firing rates. So far we have an interesting experimental phenomenon where if you perturb PV or SST interneurons, we have different synchrony patterns for temporal and rate codes. So it is very interesting, but what is the actual mechanism, network mechanism that causes these changes in iSR-IFR profile? So it's very difficult to see the actual network changes in the experimental setting with the methodologies we have. So we turn to computational modeling, especially experimental evidence suggests that PV and SST interneurons mediate inhibition through feed-forward or feedback inhibitory network. For example, if an excitatory neuron is activated to when it sends excitation to another excitation, if it activates another synapse to a feed interneuron, that could cause feed-forward inhibition. So this basically feed-forward inhibition is recruited by afferent excitation. And there's another type of inhibitory circuit motif that is recruited by itself, recurrent excitation. So this is called the feedback inhibition. So overall in the network, there is believed that there are two different types of inhibitory neural circuit motifs. One is a feed-forward inhibition and feedback inhibition. And both PV and SST, there are experiment evidence that these two types of interneurons are giving, helping these feed-forward and feedback inhibition motifs. But which one is doing what in our experimental paradigm? So we wanted to know what inhibitory network motifs do PV and SST interneuron recruit for this iSR-IFR profiles to occur, so that the synchronization of spike times at different frequencies to occur. So how do these circuit motifs are related to the perturbations that we saw using PV and SST? So we, as I said, one of the methodologies we have in our lab is to do computational modeling. And so we have, we build a model that is mimicking the cortical neuronal characteristics electrophysiologically. Sometimes we, if needed, we kind of build a full morphology computational model. But in this case, for this purpose, we only did a single compartment neural model modeling. But this is an example of how you model a full morphology computational model, and then you stack them up into a network to build a cortical column. This is not what we have done. This is adapted from Blue Brain project. But in our model, we have very single compartment computational models, each layer consisting of 200 excitatory neurons. And also within each layer, there are 50 inhibitory interneurons in each layer. And among the 50 inhibitory interneurons, there were different proportions of feed-forward and feedback inhibition. So for example, the feed-forward inhibition here provided to this excitatory neuron receives afferent excitation from the previous layer, and feedback inhibition receives intra excitation to provide feedback inhibition. And see, we modeled this as a 3-layer computational neural network model because this is most of the generalized model that's widely used. So basically, we are comparing how layer 4 neurons and layer 5 neurons are synchronized together. So intuitively, you will think that you only need 2 layers of neurons in the model, but then we don't know how many excitatory neurons, layers of excitatory neurons are actually involved in generating these synchronized spikes. So that's why we built most commonly used neural network model that has 3 layers and with an input layer. So overall, there are 200 each layer, 50 inhibitory interneurons, and then each excitatory neurons has 10% connectivity probability between the layers, because this is to incorporate the fact that there's a lot of the synaptic connectivities in the brain are very sparse. And also the input neurons, they don't receive all partake in generating inputs, but only 20% of the input layer neurons, so over 40 neurons received input. So the input we used in this computational model was recorded from layer 4 neurons in our experiment. So we feed in vivo spike pattern into the 3-layer computational network model and see how feed-forward and feedback inhibition are functioning to give rise to the synchronization of spike times across different frequencies. So when we do this experiment now, by varying the proportion of feed-forward and feedback inhibition, so it could be all feed-forward or all feedback, but as you can see here, but we vary to the ratio between them to 2:8, 4:6, etc., all the way to 10:0 again. So when we do vary the proportion of feed-forward and feedback inhibition in the network, and then do the same as the experiment, treating the input layer as layer 4 and layer 3, comparing layer 3, using the layer 3 as, for example, layer 5 or layer 6 neurons in the barrel cortex, in calculating the IFR and iSR, we see that when the ratio between feed-forward and feedback inhibition is 7:3, this can simulate the same iSR-IFR profile as in the in vivo control condition without any PV and SST perturbation. So this means this is the balanced network that allows for the synchronization of spike times across all frequency ranges when the feed-forward and feedback inhibition is at 7:3 ratio. So this is what we're going to call the balanced network to allow for these neural coding of sensory information in our work. So now using these balanced network, we're going to perturb this network by making the synaptic weights of feed-forward inhibition stronger. And to complement the increase in the synaptic weights in feed-forward inhibition, to balance the total amount of inhibition in the network, we decreased the synaptic weights of feedback inhibition so that there's a higher weight of feed-forward inhibition than feedback inhibition. So when we do that in the network and feed the same spike train and plot the iSR-IFR profile, surprisingly, this is very similar to what we see in tunnel activation of PV or inactivation of SST. And then when we do this comparison. So when we have in a balanced network, if we increase the synaptic waves of V4 inhibition, then that has a similar effect as activating PV and inactivating SST. So this means activation at PV and inactivation SST will accompany the activation of V4 inhibition in the network to make this synchronization of spike times, promote the synchronized spike times at low spike firing rate. So conversely now, if we increase these synaptic waves of feedback inhibition, then and decrease complementing it, decrease the synaptic waves of feed-forward inhibition, and then now we plot the iSR-IFR profile, we have this shape, which is similar to inactivation of PV and activation of SST. So basically, the optogenetic perturbation we did to activate SST or inactivate PV has somehow broke the balance between feed-forward and feedback inhibition in the network and promoted feedback inhibition. So overall, we have this summary. So the neurons in layer 5 and 6 are synchronized to the layer 4, coding for synchrony code for all spike times and across all frequency ranges. So these 3 different types of neural codes coexist, and PV and SST interneurons gate the synchronization as temporal codes in a frequency-dependent way. PV interneurons gating at high frequency and SST interneuron gating the spike time synchronization at low frequency. And from our experimental and computational model, we can predict that when we activate PV interneurons, that might be breaking the balance in the network, inhibitory network, to promote the feed-forward inhibition. Whereas if we activate SST interneurons, that will break the balance in the network to favor feedback inhibition. So this is one packet of a synchronized, let's say layer 4 and layer 5 or layer 6 neuron spikes. So for this synchronization to happen between the 2 spike trains for, let's say this is 12 hertz. So below 12 hertz, we have feed-forward inhibition being favored. So PV activation or inactivation of SST breaks the balance to promote feed-forward inhibition to promote the synchronization of spike times at low spike firing rates. And then activation of SST and inactivation of PV breaks the balance to promote feedback inhibition to help the synchronization of spike times at higher frequency ranges. So this study was first designed, well, not designed, but although I presented the study in the order of experiments, in vivo experiments and optogenetic experiments, and then computational modeling. In fact, this was a PhD project of one of my first PhD student in the lab, Dr. Hyun. He first made the computational model of that consists of feed-forward and feedback inhibition first for his PhD thesis. And after his PhD thesis, we wanted to find out whether this actually holds, whether feedback and feed-forward inhibition perturbation has this effect in synchronization spike times in the barrel cortex. So with the help of Professor Michael Cole from Oxford University, now in University of Glasgow, and Professor Blake Richards from McGill University, we learned, our lab, we didn't have the optogenetic methodologies before. We learned these optogenetic methods and in vivo recordings from their labs and collaborated with them together to find out that PV and SST have preferential recruitment, help the recruitment of feed-forward and feedback inhibition respectively. So in fact, although the logic of this study was penned out this way in the manuscript, in the paper, in fact, our computational model gave precise predictions on how these different neural networks might perform, encode, or generate neural codes in the barrel cortex, and that was proven by these optogenetic studies of PV and SST. So this kind of study shows how computational models are good prediction models for coming up with new experimental investigating new experiments to neural network mechanisms. So we very thank Professor Michael Cole and Blake Richards, who also helped with a lot of help with analysis of the neurocodings for the analysis for synchronization of neural codes. So Hyun Jae-jang, who did majority of the experimental works for this study, and all of the computational modeling and analysis, and with the help with Professor Michael Cole and Blake Richards, this work we were able to publish in Science Advances in last year. So now this study was kind of neat to show how PV and SST neurons activation relates to feed-forward and feedback inhibition, but there's a lot of limitation because I didn't tell you to start out with. This study was all done under anesthesia. So there's been a lot of reports that under anesthesia, there's a lot more synchronized activities. So we were able to see this kind of flat line in the iSR-IFR profile in our study. However, now if you do this experiment in awake mice, in head-fixed mice with the whiskers bundled together, now we see a completely different iSR-IFR profile. So this means that in awake state, now different populations of PV and SST as well as feed-forward and feedback inhibition will be recruited to support the neurocoding during awake state. But most interesting will be to do this experiment in awake state during active whisking. So and also what will actually happen during sensing of different texture information in awake state. So there are already a lot of studies showing when the whisker is detecting different roughness of the texture. So P100 is rough texture. The higher number means a smoother texture. And many studies show that different texture presentation to the whisker causes different fire rates in the barrel cortex. So here we are now doing further experiments to investigate what's happening in awake mice during different texture presentation of what kind of neurocodes are arising in the barrel cortex. So this is the work being done by Dr. Hyun Jae-jang and Daeono from our lab. We are presenting different textures, different roughness or smoothness of textures to the head-fixed mice and doing the same experiment, implanting an electrode to record the spike activities across all cortical layers. So we're using two different textures, P80, which is rough, and P1200, which is smooth. And now we are recording the spike activities in two different cases. And what we see is there are also, like in the anesthesia state, there are neurons that are responsive to the textures and neurons that are not responsive to the texture. So a lot of the spike trains can also be taken out for further analysis. And within these populations, the coding scheme is gone here. So across the layers, there are 2, 3, 4, 5, 6. The proportion of neurons that are selective to smooth or rough, or they're non-selective, are like this. So different cortical layers have different distributions of neurons that are selective for smooth and rough. But now if you do the same kind of experiment that we did before, activate PV or SST interneurons, this proportion of neurons within each cortical layer having different selectivity completely changes. So this is quite very preliminary data in the early days, but we will now focus on this more and see how different texture selectivity changes across layers and how PV and SST has different effects to that. And in addition to that, we are currently doing in vivo recordings using, so it's because like the success rate of recording a lot of spikes with a silicon probe is quite low. Now returning to calcium imaging using miniaturized microscopes, and especially using a lens that can monitor the calcium activities across the cortical layers, not just within the layer. So using the green lens that has this prism type, we can monitor the activities of the spiking activities or calcium activities of neurons during whisker stimulation of different texture across the layer 4, 5, 6, 2, 3, 4, 5, 6. So you can see within the whole population, the pink ones are the ones that are responsive to the texture, and the 208 neurons recorded are non-responsive. So you can see a majority of the neurons are non-responsive to any input, where small populations, like 15% are less than 15, are responsive. And within those responsive units, neurons, calcium signals, if you now split that into layer 2, 3, 4, 5, 6, different cortical layers have different responsive neuron proportions. So now we're further dividing these responsiveness into rough selective neurons and smooth selective neurons. And we will hopefully follow this on in the coming few months to analyze how these changes across the cortical layers upon PV and SST activation and inactivation. And lastly, just to show you what other ongoing projects are in the lab, we are also interested in how memories are encoded into neural codes, spike trains, and then also how PV and SST contributes to decoding of memory into spike trains. So this is our previous work. We activated PV or SST in healthy and Alzheimer's disease mice to see that specific, so for example, SST interneuron activation can restore theta oscillation impairment in Alzheimer's disease, and PV activation can specifically restore gamma oscillation impairments in Alzheimer's disease. And similar spike phase analysis show that PV is more related to gamma and SST is more related to theta oscillations. And similar, we also did spike timing dependent plasticity induction in hippocampus. And for example, in Alzheimer's disease, if you activate SST interneurons, it can restore spike timing dependent plasticity, whereas PV activation cannot. So we are using these Alzheimer's disease models to learn how the PV and SST interneurons are involved in memory processing. So this is more current work we're doing. We're trying to see how PV and SST spike patterns, codes are changing before and after learning. So we're using, in the one side of the hippocampus, we're using optrodes, and the other side using optic fiber to stimulate both hippocampi, using both PV and SST interneurons, or also using that methodologies to tag PV and SST interneurons so that we can monitor, record the spike activities of PV or SST interneurons during behavior and before and after learning. So you can see there's a light being delivered to the brain of the hippocampus. So that's activating the crimson, expressing PV or SST interneurons. And now we're gonna follow that neuron to see how that spiking activity changes before and after learning. So there should be PV and SST spike trains here, but because this is very preliminary and we are in preparation, hopefully we'll be able to upload that data in bioRxiv soon for your information later. So, and also in collaboration with Professor Michael Cole, we're also studying how the neural codes are also controlled, shaped by PV, SST, VIP, CCK interneurons in the retrosplenial cortex. So we're also doing calcium imaging using miniaturized microscopes during contextual fear conditioning. And this is excitatory neurons we're recording. And also we're doing the same with PV and SST interneurons. And also we have the tungsten wire implanted together to monitor the oscillations as well as PV and SST interneuron activity. So far we find that in the control experiments, learning somehow reduces the excitatory responses in neurons in retrosplenial cortex. So this means that reduction in excitatory frequency means that there must be some PV or SST or VIP interneurons being activated in the circuit in order to come up with these changes in excitatory signal changes after learning. So from now on, we're also trying to cross Cree and, for example, SST Cree mice and PV flip mice in order to be able to monitor the activity of two different types of interneurons to closely monitor the dynamics between one or two or even three different types of neurons in the network and how they change to generate different types of neural codes for information processing. Lastly, we are, just to introduce what we're doing in the lab as well, we are building computational network models of certain purposes. I don't want to expose too much right now, but spiking neural network, and that is sent to control a robot. So in this case, we're trying to make a robot that can navigate adaptively. So you can see it's like, avoiding boundaries already after learning. And this robot is controlled by a neural network that has spike timing dependent plasticity. And now we're trying to do this work as well. So in the lab, there's many interesting topics we're doing right now, and mostly involving interneurons, but also we're doing a lot with computational network modeling. So overall, we try to show a lot of what we're doing in the sensory information processing, how sensory information is encoded in the neural codes, and how different types of interneurons are involved in generation of these neural codes. And also, we're doing the research on how these networks are giving rise to changes in neural codes for memory information. So a lot of the most of the sensory information processing work was funded by the Human Frontier Science Program Young Investigator Award. That was the team led by Professor Michael Cole, and also shared with Professor Blake Richards, and we were able to successfully accomplish this project. And there's a lot of memory-related project ongoing in the lab. So with that, I would like to thank everybody who contributed to the work, and also thank you very much for listening to my humble talk. Thank you. I'll finish my talk here.
Thank you, Jeehyun. It was a really amazing talk, and it was really nice to see how you combine theory and experiments for this work. We have a few questions here. So thank you again for participating and sharing your work. And so let me see if I can invite people on the screen. So I will try to invite Christoforos and Aziza if you want to ask your questions. And I also have two questions from Tim. So he has to be asked offline. So one of them is, have you seen any state dependence of the synchrony, attention, alertness, etc.?
State-dependent changes in synchrony. So I briefly showed you iSR-IFR profile in our preliminary result here. So here, we actually was very optimistic when we said, okay, this is all done under anesthesia. Why don't we do in awake state? And we tried to do this experiment, and basically the synchronization level fell a lot in awake state. So and also when my Dr. Zhang told me that when this experiment was done in awake mice, unlike under anesthesia, the mice really hated having the whisker stimulated. So in 2 occasions, the electrode actually broke. The head fix was broken. So we couldn't continue with the experiment. That's why we came up with this experiment to stimulate give active stimulation of the whisker by presenting different textures instead of brute force whisking in a awake state. So obviously there are differences in synchronization level, but we were not able to further investigate that in awake state. I hope that answered some part of the question. I don't think it addresses all of it.
No, I think you answered most. Yeah, I think. But Tim also can write in the chat if you want more information. So now I'll call Aziza on screen. She also has a question
hello.
for you.
Hi.
Yeah, I wanted to ask, while you were choosing the synchrony domain, according to what you choose the border and the steps?
Oh, right. We only did I think 1 seconds of spikes. And within the 1 seconds of spikes, we saw whether the input spikes were synchronized to other spike layers.
Yeah, but it should be comparison, yes, compared to something.
Compared to something. So when you stimulate the whisker, a lot of the activities are actually occurring at the beginning, and there's an adaptation in the cortex. So that's why we didn't do that with the later spike trains. We only did it with the earlier spike trains. That, if that answers the question.
Okay. Yeah. Thank you.
Thank you. Yeah. So Christophoros Papasavas asked me to ask off his question. So his question is: the PV are typically fast spiking, whereas the SST are typically regular spiking. Do you think this difference contributes to their complementary gating of synchrony?
So that aspect we didn't get to explore in detail with our current methodology. So I just show you this. But I guess you can get some insight to what might be happening. So here in this study where they recorded PV and SST activation spike PSTH, right, responses upon whisker stimulation, you see that PV activities are, PV neurons are activated much faster. The onset is faster, whereas SST is much delayed. So this is basically causing the latency, differences in latency between excitation and PV and excitation and SST. So we think this might be one of the reasons why there was a contrasting difference in the iSR-IFR profile and also their contribution to V4 and feedback inhibition. So this is not answering your question directly. We don't exactly know how the different firing rate patterns of PV and SST interneurons are contributing to this effect, but it seems that their activation latency must be involved in giving rise to the effect we see in our experiments. And yes.
Okay, yeah, thank you. And so there is one other question from Tim Vos. He says, I would also like to know if you see differences in synchrony of the second or third spike in bursts, if they even occur in your setting.
So you mean a longer spike train?
Yeah, I think if you have like a burst, if instead of only one spike, you have like more than one, if you see differences for the second, third, and so on.
We didn't specifically see the burst patterns. We once tried to, so if you see, because this is under anesthesia, there's a lot of up and down states occurring, you see here. But we at one point wanted to, we have all the data, we haven't analyzed it yet. So there must be some effect on PV and SST activation or inactivation during up and down states where there's a burst of spike activities. But the difficulty in analyzing those data was because the onset of light stimulation was difficult to match with the spontaneous up and down states. So we once started the analysis, but didn't get to finish it. But it is a very interesting idea.
Yeah.
For needs further exploration, I think one of my students should do the analysis.
Yeah, I imagine there is a lot. I mean, if you have a big data set, there is a lot to do and to explore. So this is exciting. I have a question about the PV and SST, like the role that you compare with modeling. Like I had the impression that PV was mostly responsible for recurrent or feedback inhibition.
PV was more for feed-forward inhibition, and SST was for feedback.
Yeah, but in the circuit, how is the circuit actually in the terms of connectivity? Isn't the pyramidal neurons connected to PV and then back in the same layer?
Right. So pyramidal. So PV interneurons provide feed-forward inhibition, most possibly to the soma, and then SST interneurons provide feedback inhibition, mostly to more so to the distal dendrites. And PV interneurons are believed to have recurrent inhibition to itself as well. But that's not explored in this model. We only did feed-forward or feedback inhibition to excitatory neurons. So there's a lot of network motifs that we didn't get to consider in the network model. We just, because the sole purpose was to investigate the inhibitory neurocircuit motifs. So if you want to expand the model, there can be many dimensions to add on, but that will complicate the model. But I think it will be. Oh, another thing is we don't have any plasticity rules in our, not even short-term plasticities in our inhibitory or excitatory neuronal synapses. So that could be another limitations of our model.
Yeah. I was asking most because I from what I remember, in the canonical idea of the circuit, PV would be giving inhibitor feedback. So it's interesting that you saw, from connectivity, but that you saw that it's doing the feed forward between layers. Yeah, so that's quite interesting. So we don't have any questions. If anyone does want to have any questions, or we are now going to move to a Zoom meeting that I'm pasting here on the chat. So I'd like to thank you again, June, for this amazing talk and for answering the questions. And thank you again. It's a bit late for you there, so thank you for taking the time to.
My pleasure to be able to share my research here. My honor.
Thank you. And then, so let's move to the Zoom meeting. So the link is in the chat. Please, whoever wants to join us, just you can join us there. And thank you again. And bye-bye, everyone.